I want to join other speakers to really express my gratitude to the organizers of the meeting, Xin and Yajing, and they have done a terrific job, as always, to bring the best science in the Great Ball and AHS. And it's I've been actually involved in this organization for many years, and I really so proud to see the great progress and incredible advancement in the field in China. And today I want to share with you some of the relatively recent findings and outcome from our study that targeted to metabolism for HFpEF. And first, I want to give credit to my incredible team, both at UCLA, but also particularly people from Duke and US and in Singapore. And as we know that HFpEF is actually an emerging new challenge in heart failure, and it's getting more prominent in the world is because of the prevailing increase of the metabolic disorder underpinning many of, and as well as aging, underpinning our human population. So right now it's almost about 50% of people that actually suffer from HFpEF. And the challenge is that those diseases essentially have a very different ideology compared to the so-called traditional heart failure that triggered by either ischemic or mechanical overload. And so we, as a basic scientist, always enter those questions. It's a very simple-minded approach, but in the past, we always treat the heart as really a sports car, mechanically very strong because it's need to pump blood, right? Because every day we have to pump almost 2,000 gallons of blood for a normal human heart. But we often overlooked the importance of the metabolism or energy consumption, and as I think illustrated by several other talks today, is that this heart is also a high-performance sports car that require a very different kind of fuel. Indeed, every day our heart, the normal heart, actually consumes almost 6,000 grams of ATP if you just calculate the molecule weights, and that's of course exaggerated number because the energy we extract from the ATP is not the whole molecule, but only the final phosphor sort of energy that in the last phosphorylated bond. And so why that is very important is because almost all the previous traditional therapy is actually targeted to the mechanical problem of the heart, unloading, increased contractility or reduced contractility. And much of that is almost like you fix the car in the workshop, right? You have a car broken. Okay, let's take the car out and to the garage and then just mechanically try to figure out what is the problem. And then unfortunately, don't worry about the small fund. It's just all those traditional therapeutic targets that apply to HFpEF actually generated neutral or basically no effects. Right? So that actually requires us to think the heart failure, especially HFpEF, in a sort of new angle. And that's probably why a couple of years ago, the AHA and the National Institute of Heart and Lung and Blood in the United States put up this white paper and calling the HFpEF is the most greatest unmet need in cardiovascular medicine today. And, in American, we usually don't use these words very often. And to actually have a consensus, our community, to recognize is the greatest is actually quite rare. So we know that this is really a big problem. However, the glimpse of the hope is that some of the drugs actually never intended to heart failure actually turned out to be beneficial. This is SGLT2 originally in the outcome study for a diabetes drug and find that they actually can reduce cardiovascular incidence for more than 30%. However, there is no improvement of mortality. And so that still shows there's still perhaps a long way for us to figure out what could be there's a lot of more room for improvement. But nevertheless, the mechanism of HFpEF is still very complex. There are many, many ideologies. It's not a single disease. It's actually a spectrum of the disease. It could involve many other organs, as illustrated in this very nice sort of outline. So when we investigated the experimental models, and we often found that the single stress usually do not generate a very uniform or clear-cut sort of a HFpEF phenotype, and that's perhaps why in the current days, most of the HFpEF models are sort of a multi-hit or second-hit model. And so our lab actually also tried to, early on, try to understand the disease is never actually think very carefully about the HFpEF at that time. It was quite a few years ago. We were simply curious about how the metabolic stress and the mechanical stress interact together. And so we put this pressure overload model in a regular mouse and in the purple, and also in this blue mouse, which is actually the OBOB mice. And at that time, we totally expect that the OBOB mice is going to get a lot worse in terms of heart failure based on the literature. But what we observe is actually at the beginning quite opposite. If you look at the red, the ejection fraction actually remain constant even with after 10 weeks of pressure overload. And yet the traditional sort of lean mice developed a very classic heart failure with reduced ejection fractioning. However, they are not really any better. Turned out those mice actually developed more severe diastolic dysfunction, as we demonstrated from this parameters of echo EE prime, and also from the strain analysis and showing the relaxation impact or index is impaired. And so we actually demonstrated this diastolic dysfunction using the the invasive hemodynamic method, and support our earlier observation by the pressure overload by the echo, and we clearly see that those animals also developed end-diastolic pressure higher and also relaxation defects. And beyond that, if you look at histology, they are much bigger in heart, so hypertrophy is actually worse. And you can see that they're both the cardiomyocyte hypertrophy as well as pressure overload. I mean, the remodeling associated with the marker genes are significantly elevated, especially in the inflammatory genes. However, the traditional markers like BMP and actually not actually are reduced, and that's in fact quite similar to what we observe in the clinic as well. So the traditional markers of heart failure do not really reflect well with the HFpEF. We indeed observe oxidative stress in those animals and associated with, again, the inflammation and the fibrosis. So their heart is not any better. And so we were facing this kind of interesting phenomena where the heart, if it's a normal heart, under mechanical stress is the so-called HFrEF. But if you go through this obesity and diabetic state, they actually generate a new form of heart disease similar to what we observe in the clinic called HFpEF. And so we, as a basic scientist, again, we were trying to understand what could be the underlying mechanism. We use single cell studies to actually dissect what could be the signature among different cell types. And today I have time to just focus on the cardiomyocytes. If you take out the normal cardiomyocytes and after pressure overload, they developed classic HFrEF in this very different, very separated molecular state. So that's actually well known. And when we look at under the OBOB condition, this is diabetic obesity condition, this heart, even at the baseline before pressure overload, already separated. So they clearly started from a very different place. But after pressure overload, you can see that they are stuck in this middle. They never go through the same sort of a disease progression as the normal cardiomyocytes. And so that gave us, again, the evidence that this is probably a different disease from the HFpEF is a really different pathogenic process as the HFrEF. And when we look at those genes in the cardiomyocytes, compare the HFpEF and the HFrEF, and try to understand what is the major pathways affected by those genes. And it's not surprising metabolically. It's the number one is the insulin. The second, surprisingly, is actually the glucagon signaling. And we can demonstrate that by chemically, but molecularly, it doesn't make a lot of sense because glucagon is supposed to be activated or express. The receptor itself is expressed mostly in the liver, and we know the pancreas, and the most of the impact is supposed to be the metabolic regulation, right? It's in the glucose and the insulin response. But there are anecdotal evidence already supporting that both insulin signaling and the glucagon signaling perhaps are very important for the physiology and function disease progression in the heart. And so we performed the first experiment is using this REMD compound, which is actually a glucagon neutralizing antibody, basically to block the glucagon signaling. And when we apply that before we start the pressure overload, you can see that the study shows that by preventing the glucagon activation, you can almost completely block the hypertrophy process in this HFpEF condition, and also reduce the oxidative stress, and also essentially restore or prevent many of the manifestation in terms of molecular as well as histological features. But you would argue that those global changes, because of you improve the systemic metabolic health. So we did this experiment is using the glucagon receptor specific knockout in the heart. And when we applied this study, we for sure, actually, I was remember, when we discussed in the lab meeting, I said, for sure, this is going to be a negative result because we essentially to say, there's no expression in the glucagon receptor in heart or very low. And so the impact could be minimal. And turned out I was wrong. And the glucagon receptor knockout in the heart turned out to be protective against the cardiac dysfunction, especially diastolic dysfunction in the heart under both the high-fat diet plus pressure overload condition. So this is, again, suggests that there is a cell autonomous effects of the cardiomyocytes in response to the elevated glucagon in the diabetic condition, and that contribute to this HFpEF phenotype. So if that's the case, whether this particular observation can be applied to more generic HFpEF models. And so this is actually the Joe Hill model from Dr. Hill's lab and published in Nature using the high-fat diet plus LNM. And we use the system to essentially test the glucagon antagonist. Instead of applying now the glucagon, the glucagon neutralizing antibody at the beginning, we actually applied 8 weeks after the onset of the stress, and make sure that this has actually any therapeutic sort of impact. And what we observe is that yes, we see both the metabolic disorder as well as the hypertrophy, and most strikingly is this nitrosylation impact are very significantly removed. And so that gave us, again, the concept that glucagon receptor antagonist could be an interesting therapeutic target. And knowing that glucagon receptor is now this particular neutralizing antibody is already in phase 2 for diabetes. We thought that this could be a very nice mechanism for us to repurpose the drug for HFpEF therapy. And again, in the same token, when we actually studied the HFpEF model, and we performed a metabolic profiling, and actually that led us to the discovery of the major change of the branch amino acid. As we know, this is essential amino acids, and most of the people actually can use this amino acids to actually do the bodybuilding and eat a lot. And what we see is actually in the heart, both the HFrEF in the left and also HFpEF, published by David Katz's lab, the BCAA defect is a very prominent molecular signature at the gene expression level. And we also performed the metabolomic studies in the HFpEF versus a normal human population. And we observed a different modules of metabolite associated with the different pathological outcome, and especially with the yellow module, which is highly associated with the very severe adverse outcome among the people, patients. And when we decode what is the constitutes in this yellow module, they are prominently featuring the branch amino acid and the glucose. So this is an unbiased approach to actually identify BCAA metabolism could be a major problem. So we performed in the preclinical model of this HFpEF. This is actually in Joe Hill's model again, and showing that in this model we can reproducibly generating this HFpEF phenotype. And what we saw was that the BCAA so this is just again to show that we can fulfill all the requirements actually published by Joe Hill in this model. And what we observe is that the BCAA metabolic pathways are impaired, and also with the accumulation of the metabolites as we expected. And so we use the BDK, which is an enzyme that actually can restore the BCAA catabolic activity, as a genetic tool to demonstrate that if you restore this BCAA flux, you can actually completely reduce the metabolite accumulation. And this model is actually very protected against diastolic dysfunction as well as the remodeling process in the HFpEF condition. And we also observed that they can restore the, at the mechanistic level, the insulin sensitivity as well as the MPK activity. And we were actually very excited to see that the hallmark of nitrosylation in the protein was very significantly restored, but not as much as the pressure, the blood pressure change, as actually earlier demonstrated by Zolt Arani's lab. So we think the real mechanism is not the pressure, blood pressure change, but actually it's on the intrinsic property of the cardiomyocytes. And to demonstrate the therapeutic impact, we also use a BDK inhibitor, it's BD2, to apply that to a well-established model where we actually, again, using almost 10 weeks of pressure, the high-fat diet and LNM treatment, and you can see that we can restore the both hypertrophy as well as the metabolic changes and diastolic dysfunction, and suggesting that this could be a very viable therapeutic approach. And this is just using the hemodynamic to see the same. So again, this is the kind of our mechanistic scheme. And the interesting or exciting part is that exactly the same target now is in clinical trial for phase 2 HFpEF by Pfizer based on our study. And so with that, I share with you the two potential additional metabolic targets in addition to the SGLT2 or the GLP-1 that contribute potentially to the HFpEF pathogenesis as well as therapeutic targets. And I want to acknowledge our collaborators and the funders. Thank you so much.